Hi guys, Mrs. Spurlock here. This week we are covering Chapter 5, Lesson 5, Advances in Genetics. Now, before everyone freaks out at me at the same time, I'm aware that the first page of the packet is missing. I don't know how I skipped scanning it. Don't worry about it. Your life will go on. I promise. So, we only have one big question this week, and that is how can organisms be produced with desired traits? The first method of producing organisms with desired traits is called selective breeding. This happens when basically a human gets to decide, I want this dog and this dog to have puppies because I like the length of this one's coat and I like this one's temperament. Does it work exactly the way they want it to 100% of the time? No, but it's a method that's been used for a very, very long time. Uh, there are two ways to selectively breed for traits. The first way is called inbreeding, and that's where you take two organisms that are closely related and allow them to create offspring. The dangerous thing about this is if you look at the PowerPoint, this top picture demonstrates what inbreeding can cause over a long period of time. On the left, you have a picture of what bull terriers looked like uh, very long time ago, and over the years, because they've been inbred to keep the same traits, to keep the traits pure, you can see that they've developed this singular, almost triangle shaped head, and they often have problems with sight because of the way their eyes are positioned on their head. The other almost opposite process is called hybridization, where you cross two genetically different individuals. Now, this image at the bottom here, this is called a mule, and a mule is a hybrid of a horse and a donkey. And the goal there is that the animal has the strength and endurance of a donkey, but is a little bit more obedient like a horse, because the stereotype of the donkey is that they're very stubborn. And so this hybrid kind of solves both problems, but hybrids are infertile. They can't have their own offspring. So it's not a new species, but it is a combination of two different species. The next way that organisms can be produced with desired traits is via cloning, where an organism that has the exact same genes is produced from an organism with identical genes, right? You guys have heard of cloning before. The image in the background here is of two puppies that have been cloned. You can get your pets cloned, but I believe it costs about $150,000. So none of us will be getting our pets cloned anytime soon, probably. And the last way that we can produce an organism with desired traits is through genetic engineering. And this occurs when genes from one organism are transferred into the DNA of another organism. We're going to talk about the way that genetic engineering happens in bacteria.
Bacteria have a really special quality that makes them excellent candidates for genetic engineering, especially genetic engineering that makes drugs or hormones for humans. And we're going to talk about why right now. So, we're going to talk about the way that we get bacteria to make insulin. Insulin is a hormone that people who have diabetes do not naturally produce, so they rely on insulin supplies primarily from bacteria to keep them alive. So in this bacteria, this black blob here is going to be our bacteria. And inside of the bacteria, you're going to see this red ring. That red ring is a special ring of DNA called a plasmid. That's not the only DNA they have. Bacteria also have a much longer strand of DNA where most of their genetic information is found. And that's going to be right here. It's bacterial chromosome. Scientists take this plasmid out of the bacteria and they use an enzyme to cut it open. At the same time, they're using the same enzyme to cut this strand of human DNA. This purple piece here is also human DNA. So it's not separate from the other DNA. It's just that after the enzyme came in, it made cuts right here and right here. And so now you've got 3 pieces of DNA. Well, these 2 pieces are going to come together because they were cut with the same enzyme. And they're going to create a new plasmid that didn't exist before. And now this plasmid has the gene to create human insulin. Well, the plasmid isn't going to do anything on its own outside of the bacteria, so then the plasmid, with its new recombinant DNA, is put back into the bacteria. And so now it's doing bacteria things, but also creating insulin and reproducing to create new cells that also have the genetically engineered and normal DNA in them. And so one of the really huge benefits is that by allowing this bacteria to reproduce over and over again, scientists end up with exponentially increasing numbers of bacteria to create the insulin. So instead of just having one bacteria that's making all the insulin, that bacteria is going to reproduce and create more bacteria and more bacteria and more bacteria, all of which can also create insulin.
Now, in other organisms, because they don't have plasmids, you can only insert the DNA directly into the gene. That means you have to do this when the organism is a single cell or just a few cells. You can't wait until the organism is made of hundreds or thousands or millions of cells. A really unique benefit to genetic engineering is something called gene therapy, where doctors have developed the ability to create a medicine where, when it is applied to the cells that have a certain disease, it kind of solves the disease. So, for example, there is a genetic disorder called cystic fibrosis, where a person's respiratory system produces too much mucus and they can't breathe very well. It's all real gunky and fluidy. And so through gene therapy, they can inhale in an inhaler this genetically engineered bacteria that it's actually a virus that goes in and infects the cells along their respiratory system. But it doesn't infect them and cause a disease or a sickness of any kind. It infects the cells and allows them to produce whatever they need to produce normally. It allows them to behave normally. Now, when those cells die, when they divide, they don't pass on the new gene to the resulting cells. When those cells die, the gene therapy is gone. And so gene therapy is something that has to be done repeatedly throughout the person's lifetime. And there's a really valid concern that there's no way for us to know the long-term side effects of genetic engineering. So it can be a bit of a tense subject in some situations. And those are the 3 ways that we can produce an organism with desired traits: selective breeding via inbreeding or hybridization, cloning, and genetic engineering. All right, see you guys next week.